Hello, my name is Meson Cheng, and my partner's name is Elias Smith, and today we will be explaining the cell cycle, specifically the process of meiosis. Meiosis is the division of cells that produces four haploid cells. Unlike mitosis and binary fission, meiosis does not create clones, but four genetically distinct haploids. Thus, only an organism that can sexually reproduce can undergo meiosis. Before the cell goes through meiosis, they must first be prepped and go through interphase. During interphase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into the meiosis phase, many internal and external conditions must be met. The three stages of interphase are called G1, S, and G2. The first stage of interphase is called the G1 phase, or known as the first gap, because from a microscopic aspect, little change is visible. During this phase, the cells grow, develop, and differentiate. The chromosome complement in this phase is 2n1x, meaning each chromosome has only one chromatid. Before moving on to the next phase, cells go through the all or nothing checkpoint, where the cell will be committed to dividing after passing through it. However, if the cells do not pass through the checkpoint, the cells will enter G0, where cells have lost their ability to divide, like muscle and nerve cells. The next stage is the S phase, where DNA synthesis takes place. The S stage of the interphase takes the longest because of the complexity of the genetic material being duplicated. In the S phase, DNA replication results in the formation of identical pairs of DNA molecules. Sister chromatids thus having the chromosome complement of 2 and 2x, where there are two centromeres and two chromosomes. The sister chromatid will then be firmly attached to the centromeric region. The last stage of interphase is the G2 phase, where the cell gets ready to divide by making proteins, storing food, and replicating organelles. The cell does this to replenish the energy stores and to synthesize proteins for chromosome manipulation. There is also additional cell growth during the G2 stage, and the final preparations for meiosis are completed before the cell is able to enter. Now that the cell has gone through interphase, it is now prepared for meiosis. There are two stages of meiosis, meiosis 1 and meiosis 2. During the first stage of meiosis, homologous chromosomes are broken down. It is the goal of turning cells from diploid into haploid and choose the chromosome number. In the second stage, sister chromatids are broken down into individual cells. Meiosis 1 starts with prophase 1. In prophase, the chromatin located inside the nuclear membrane of the cell condenses to form chromosomes. The pairs of chromosomes created are called sister chromatids, and they are attached to one another at the centromere. On these sides of the cell, the mitotic spindle creates microtubules. Spindle fibers are then created, which creates a spindle apparatus. Synapsis then occurs. Synapsis is the process in which the homologous chromosomes stick together in pairs as four chromatids, known as a tetrad. During synapsis, chromatids can overlap and fuse, also called crossing over. Crossing over recombines sections of DNA to create new and random combination of genes. The nuclear envelope also disintegrates. The centrosomes then start moving to opposite poles, and then the mitotic spindle form appears and attaches the chromosomes at the centromere. The place where the mitotic spindle attaches to the centromere is called a kinetochore. After that, metaphase 1 occurs. During metaphase 1, the tetrads begin moving towards the metaphase plate and eventually are lined up at the metaphase plate, which is the middle of the cell and are lined up by one microtubule. The law of independent assortment is that the orientation of one pair of chromosomes is independent and indifferent to the orientation of another pair of chromosomes located on the metaphase plate. This acts as a source of genetic variation for the cell. After that is anaphase 1. During anaphase 1, each homologous pair is attached to a microtubule. During this phase, the homologous pairs are broken apart and are separated to move towards opposite sides of the cell. To do this, the microtubules in the spindle apparatus will separate the sister chromatids, while the kinetochore microtubules shorten and non-kinetochore microtubules lengthen. As the microtubules push the pairs apart into opposite parts of the cell elongates, setting up telophase 1. During telophase 1, the parts are separated and are at opposite ends of the cell. The cleavage furrow forms inside of algal and animal cells. The nuclear envelope also reforms. Telophase and also starts with cytokinesis 1. The process occurs in the midline of the cell. It separates the cell into two cells that the centrosomes start to move towards poles of the cells. After, either oogenesis or spermatogenesis splits the cell. After this, there is another round of meiosis called meiosis 2. During this, the two cells made from meiosis 1 are then split into two haploid daughter cells. Like meiosis 1, meiosis 2 starts with prophase 2. During prophase 2, things that happened in prophase 1 happen all over again, except for synapses and crossing over. The cell membrane surrounding the nucleus in each daughter cell goes away. Like in prophase 1, the loose chromatins are condensed to chromosomes. Centrosomes start to move towards poles of cells. The centrosomes are duplicated, and the spindle apparatus is recreated. The nuclear envelope breaks down again. The mitotic spindle forms again and attaches the chromosome to the centromere. Next is metaphase 2. Like in metaphase 1, the sister chromatids start moving towards the midline of the cell, also known as the metaphase plate, and line up along it. They are lined up individually along the metaphase plate. Each sister chromatid is then attached to a microtubule. The law of independent assortment still applies, stating that different pairs of chromosomes segregate independently of each other. Next is anaphase 2. In anaphase 2, sister chromatids of each chromosome are separated and move towards opposite parts of the cell. The sister chromatids are now at separate ends of the cell. In telophase 2, the nuclear envelope appears and the spindle spirals disappear. In cytokinesis 2, each cell is separated into two cells, creating 4 haploid daughter cells. Either eugenesis or spermatogenesis splits the cell. In eugenesis, 3 polar bodies and 1 viable cell are produced. In spermatogenesis, 4 viable sperm cells are produced. In the end, thank you for listening, and we hope you enjoyed our presentation.